When you find a mollusk shell on the beach, maybe a spiral or just the smooth inside of an oyster shell, you see this incredible beauty and strength,
Yeah.
or resilience. They feel so solid.
Yeah, remarkably so.
But how do these creatures without bones or, complex tools, how do they build structures this intricate, this durable? How do they craft their homes almost atom by atom, layer by layer?
It really is one of nature's truly spectacular feats of engineering. And, well, that's exactly what we're diving into today.
Welcome, everyone.
Welcome to a deep dive from the All Sorts podcast, your guide to uncovering the fascinating, often hidden science behind the natural world. We are exploring the incredible architecture of mollusk shells and the sophisticated biological process that builds them called biomineralization.
And this deep dive comes straight from the sources you've shared with us, a collection of scientific research, including papers that get right down to the molecular level. They're looking at specific proteins involved, exploring the microscopic structure of different shell layers, and examining how these amazing natural buildings are holding up against challenges like, well, environmental change, specifically ocean acidification.
Yeah, that's a big one. So our mission in this deep dive is to cut through some of that complexity, pull out the key insights from this material, and really try to understand the essential building blocks and processes behind shell formation.
And also see the real-world stresses these structures are facing now. Let's get started.
Let's do it.
So, okay, let's start with the basics. What are shells actually made of? I mean, it feels like rock, but it's not quite that simple, is it?
No, not at all. That's where it gets really interesting right away. Mollusk shells are remarkable composites. Their main ingredient is calcium carbonate, KCO3. But here's a key insight. This isn't just one uniform material.
Ah.
Calcium carbonate can crystallize in different forms known as polymorphs.
Polymorphs, like different shapes or arrangements of the same basic stuff.
Exactly that, yeah. The sources highlight the main ones we find in shells: aragonite, calcite, and sometimes vaterite. Different species or even different parts of the same shell will use different polymorphs.
Okay.
Think of it like choosing different types of bricks, maybe, for different parts of a building, depending on what you need that section to do. Strength here, maybe something else there.
Okay, so you've got the mineral bricks, calcium carbonate in different forms.
Yeah.
But they aren't just like piled up randomly. There's something else holding it together, guiding how it's lowered down.
Crucially, yes. This is the other absolutely essential component. The organic matrix.
Organic matrix.
It's a complex network made of proteins and polysaccharides like chitin embedded within those mineral layers. And this isn't just passive glue or filler.
Yeah.
No, the organic matrix is really the active blueprint and, you could say, the construction crew.
So it directs the whole operation.
Yeah.
Tells the minerals where to go.
Precisely. These organic molecules control the exact location, the orientation, the size, and the shape of the mineral crystals as they form. Without this matrix, you'd probably just get a lumpy mineral deposit.
Right, just a blob.
Exactly. With it, you get the stunning, highly organized, functional structures we see in shells.
Which brings us to the architecture itself. Because different mollusks have radically different shells, flat oysters versus those intricate spiral snails. And even within one shell, there are different layers, different designs.
Absolutely. The sources describe several key microstructures, the sort of internal architecture. One you definitely know is the nacreous layer, or mother of pearl.
Oh yeah, beautiful stuff.
It's not just beautiful with that shimmer, that iridescence, it's incredibly tough. Structurally, it's built like a microscopic brick wall. You've got stacked layers of these tiny aragonite platelets.
Okay.
With thin sheets of that organic matrix sandwiched between them.
That layered structure, the sort of brick and mortar at a tiny scale, that must be key to his properties. Even if the sources don't detail exactly why it's so resilient mechanically, that stacked design sounds fundamental.
It absolutely is. Then there's often the prismatic layer. This is usually the outermost part, and it's made of elongated calcite crystals, typically oriented perpendicular to the shell surface.
Perpendicular, so like columns.
Yeah, think of closely packed columns standing upright.
Right. Columns may be protecting the nacre layers underneath.
Could be part of the function, yes. And then a particularly complex one found in some snails and clams is the crossed lamellar structure. This involves really intricate interlocking laths, like tiny planks of aragonite or calcite.
Wow.
Arranged in opposing directions across different layers. It's sort of like nature's plywood or maybe even reinforced concrete with rebar. Provides amazing resistance to fracture.
It's fascinating how nature uses these different arrangements for different needs, like building walls, columns, or these woven structures. And the sources mentioned scientists use some pretty cutting-edge tools, right? Like microscopy techniques like SEM-EBSD and FibTem
Exactly.
to actually see and analyze these tiny nanoscale designs.
That's right. They can peer right into the shell's internal structure at a level of detail that really reveals this incredible biological engineering. But okay, how do mollusks do this? How do they execute this engineering with such precision?
Right. How does it work?
That's the work of the molecular architects.
Biomineralization in action.
Yes. The key tissue responsible is the mantle, which lines the inside of the shell. It's like the skin right up against the shell.
Okay.
That's where the shell material is secreted from. So scientists study this by analyzing the proteins found within the shell layers themselves and also in the mantle tissue. And one of the sources dives really deep into a specific example.
Yeah, it really highlights the scientific detective work involved in figuring out just one piece of this enormous puzzle. They look at a protein called HIC22 in a freshwater mussel. Is that right? Hyriopsis cummingii.
That's the one, source one. The core insight here is identifying specific proteins within the nacre layer, the mother of pearl, that are likely directing how it forms.
Okay.
They isolated this protein, figured out its size about 22 kilodaltons, which is a measure of molecular weight, and they determined its N-terminal sequence. That's the beginning part of its amino acid chain.
Like getting the first few letters of a very, very long word.
Exactly, a unique identifier. And using that information, that sequence tag, they were able to hunt down and find the gene that actually produces HIC22.
Clever.
Then, a really crucial step was figuring out where in the mussel's body this gene is active. Is it everywhere or just in specific places?
Right. Is it involved in shell building or something else entirely?
Precisely. So using techniques to visualize gene expression, basically seeing where the gene is turned on, they confirmed that HIC22 is indeed produced in the mantle tissue and also in the pearl sac.
The pearl sac.
Yeah, the specialized bit of mantle that makes pearls, which are essentially nacre. So it's made in the very places responsible for building the shell's nacre layer.
Okay, so the finding is, here's a specific protein, HIC22, made exactly where nacre is formed. What do they think its job is? What does it do?
Well, the initial findings suggest HIC22 is what's called an acidic matrix protein. These types of proteins, rich in acidic amino acids, are thought to be crucial for controlling the deposition and organization of calcium carbonate in nacre.
How so?
The source indicates it likely plays a role in directing how those aragonite platelets, those microscopic bricks, are laid down in that regular, ordered way. They interact with the calcium ions or the growing crystals. More research is definitely needed to pin down the exact mechanism.
Sure.
But identifying specific proteins like this is a huge step in understanding the molecular machinery of nacre formation.
And HIC22 is just one player in this molecular orchestra, right? There must be others.
Oh, absolutely. Source 2 mentions several others that have been identified. Proteins with names like asparage, which seems to influence crystal shape, particularly in the prismatic layer. Perlucin, which can help start or nucleate crystal formation.
Okay.
PIF, which is known to be a key player in nacre formation involved with aragonite. And prismalin 14, found in the prismatic layer, as the name suggests.
Lots of specific molecules.
Yeah, and the key takeaway here is that different proteins are found in different shell layers, and they each likely have very specific jobs in guiding that mineralization process. Controlling which polymorph forms calcite or aragonite, the crystal structure, how fast it grows, how everything is precisely organized into those layers we talked about.
It's really incredible to think that the strength and the beauty of a seashell comes down to this intricate dance of mineral deposition, all guided by specific proteins operating at the molecular level. Just amazing. But even with this incredible biological engineering, shells face some significant challenges today.
They absolutely do. And this brings our deep dive to a critical environmental issue. The sources discuss the impact of ocean acidification.
Right.
As atmospheric CO2 levels rise, mainly from burning fossil fuels, the oceans absorb a lot of it. And this chemical reaction leads to a decrease in the ocean's pH, basically. The water becomes more acidic.
And the big concern there is how this affects creatures that build shells made of calcium carbonate, like molluscs.
Exactly. The leading hypothesis, the main idea, is that the lower pH reduces the availability of carbonate ions. These ions are the fundamental building blocks mollusks need to combine with calcium to form their calcium carbonate shells.
So it's like trying to build a brick house when bricks are suddenly harder to find or more expensive.
It's a pretty good analogy, yeah. It can make it harder and certainly more energetically costly for them to calcify to build their shells. And in more extreme conditions, it can even cause existing shell material to start dissolving.
That sounds worrying. So what do the studies cited in the sources actually show is happening out there? Is it just a hypothesis?
No, unfortunately, it's more than that. Source 3 provides a synthesis, kind of a summary, of many, many studies, and the overwhelming finding from most of the research is, well, negative.
How negative?
A large majority of studies report significant negative effects on mollusk calcification and growth. This includes things like slower shell growth rates, reduced shell thickness, weaker shells, and sometimes visible dissolution or pitting on the shell surface.
Wow. Are there specific examples? Which species are affected?
Yeah, the source mentions studies on species like the eastern oyster, Chrysostria virginica, the hard clam mercenaria, the soft clam Emea arenaria, the predatory whelk Urosalpinxenaria, the periwinkle Littorina littoria, the conch Stombus alatus, and the bay scallop Argopectiniradians. A lot of commercially and ecologically important species have shown these negative responses in experiments.
That's a concerning list, but is it always negative? You mentioned most studies. The sources mention some variability, don't they?
They do, and that's one of the key and perhaps more complex insights from the research. The picture isn't entirely uniform. While most species studied show negative impacts, some studies, like one mentioned on the slipper limpet Crepidula fornicata, have occasionally shown maybe neutral or even slightly positive responses at certain moderate CO2 levels.
Really? Positive.
It's not consistent even within limpets and often disappears at higher CO2 levels. And interestingly, cuttlebone calcification in cuttlefish, that internal shell, sometimes shows neutral or even positive responses in lab experiments, too. So it's not a simple acid bad for all shells story.
Okay, why this difference then? Why are some species or maybe even populations seemingly more affected than others?
Yeah, that's the big question. The sources point to several potential reasons for this variability. It could relate to the type of calcium carbonate polymorph they use. Aragonite, which forms nacre, is generally thought to be more soluble, maybe more vulnerable than calcite, though the reality is complex. Their ability to regulate their internal acid-base balance is also likely a major factor. Some species might just be physiologically better at buffering themselves against external pH changes happening in the seawater.
Makes sense.
And significantly, the environmental history of a particular population seems to matter. Organisms from populations that naturally experience more variable pH conditions, maybe in estuaries or upwelling zones, might have a greater inherent capacity for adaptation, or what's called phenotypic plasticity, basically, being flexible in their response.
So some might just be better equipped to handle the change based on their evolutionary past or their environment. What about different life stages? Are like baby mollusks particularly vulnerable?
Yes, the sources definitely highlight that. Early life stages, especially the larva like the tiny swimming trochophore stage when shell formation first begins, can be extremely sensitive.
Why them specifically?
Well, they're undergoing rapid development, forming their first shell, and maybe haven't developed the robust physiological regulation systems that adults have. Studies show elevated CO2 can lead to decreased calcification, reduced survival rates, and slower development in larvae of many species.
And do we know why that happens at a deeper level?
The vulnerability might be linked to disruption in energy metabolism, changes in gene expression, maybe the genes needed for shell building aren't turned on properly, or difficulty synthesizing those crucial shell-forming proteins we talked about earlier.
It sounds like a really tough challenge for them, trying to make their first protective shell when the building blocks are harder to get and maybe their internal systems are stressed.
It really is. And the sources also note that for some species, even adults, coping with elevated CO2 seems to involve an increased metabolic cost. Meaning they have to expend more energy just on basic bodily functions like maintaining their internal balance. That potentially leaves less energy available for things like growth or reproduction.
So even if they survive, they might not grow as well or reproduce as much?
Exactly. And this response also varies. Sometimes, depending on other environmental factors like temperature-combined stresses can be worse. And importantly, these lab findings are backed up by field studies near natural underwater CO2 vents, places that mimic future ocean conditions.
What do they find there?
They observe things like reduced populations of young bivalves and gastropods settling, and reduced shell strength in adult snails living closer to the vents where the pH is lower. It confirms these effects happen in the real ocean, not just the lab.
So wow, we've journeyed from the basic mineral ingredients, the KCO3 polymorphs, and the intricate architecture of mollusk shells.
Yeah, the nacre, the prisms.
Delved into that unseen world of molecular architects like HIC-22 that guide this process with such incredible precision.
The organic matrix directing everything.
And it landed on this very real, very current environmental challenge they face from ocean acidification, making it harder for them to build those amazing structures.
It really is a profound illustration, isn't it? The complex interplay between biology, chemistry, geology, and the environment. And it's a testament to the sophisticated natural engineering happening all around us, even within these seemingly simple creatures like snails and clams.
Absolutely. And just as mollusks meticulously build their remarkable shells layer by layer, molecule by molecule, your understanding of the world is built piece by piece, insight by insight, deep dive by deep dive.
Well said. And if this exploration into the incredible world of mollusk shells sparked your curiosity, if it added a valuable layer to your own knowledge structure, you can continue building that understanding and help others discover these insights too.
You could do that by liking this deep dive. Think of it as helping the insights calcify for others. Share it with someone you know who would find this fascinating, maybe someone who loves the beach or is interested in biology or materials science. And please subscribe to the All Sorts podcast channel on YouTube.
Yeah, every like, share, and subscribe genuinely helps our community of curious learners grow and helps us keep diving deep into the world's fascinating topics, bringing you insights from sources like these.
Remember, we've already crafted other deep dives on a really wide range of topics, just waiting for you to explore. Maybe check out the one on mention-related topic if applicable, otherwise general next.
Dive into those as well, and you can stay connected with us and discover new areas for exploration by following the All Sorts podcast on X, Instagram, and Threads.
We'd love to see you there.
Definitely. So as we wrap up, maybe consider this final thought. The incredible resilience, but also the varied responses of mollusks to environmental stress, like ocean acidification, highlighted in these sources, they offer real insights into nature's adaptive capacity, but also its limits. How critical is it really for us to understand these fundamental biological processes, from the molecular architecture of a shell to the physiological stress response as our planet undergoes such rapid environmental change? What other hidden mechanisms in nature, what other molecular toolkits hold keys to survival in a shifting world just waiting to be uncovered, perhaps layer by intricate layer, just like a mollusk shell?